The arrival of COVID-19 vaccine is likely the most awaited item in 2021 for many people, including Malaysians. As of January 2021, the Malaysian government has procured vaccines covering 82.8% of the population to be administered in 2 doses, in hopes of achieving herd immunity, with brands from Sinovac, Pfizer-BioNTech, CanSino Biologics, Oxford AstraZeneca, Sputnik V and WHO COVAX facility. However, what are the different types of vaccines? To achieve immunity from a virus, the body must have sufficient levels of neutralizing antibodies that recognize the virus. This process, called adaptive immunity, is generally the same for all vaccines, beginning with the presentation of viral antigens to immune cells. Antigens, or antibody generators, are particles on foreign pathogens that induce an immune response, leading to the production of antibodies. In the case of COVID-19 virus, the most targeted antigen is the distinctive spike protein of the coronavirus, which helps the virus to infect host cells. Upon the presentation of spike protein antigens by antigen-presenting cells to helper T cells, then to plasma B cells, neutralizing antibodies specific to the spike protein will be generated, along with memory B cells, which is vital for long-term immunity. The differences between vaccines lie before the antigen presentation step. The first type of vaccine is inactivated, or killed virus vaccine. This is a traditional method of making vaccines, with examples such as polio and rabies vaccines. It works by injecting inactivated COVID-19 virus into the body, stimulating immune cells with its spike protein, while not causing harm to host cells through viral replication. This method is applied by Sinovac. The second type of vaccine is viral vector vaccines, specifically using adenovirus. Adenovirus is a common cold virus that causes mild respiratory disease. However, through genetic engineering, recombinant adenovirus can be produced with non-replicating properties. Hence, it becomes merely a harmless shell, called vector, to effectively carry genes to host cells. This is done by deleting the E1 gene from their genome. Additionally, the E3 gene is also deleted, in order to allow larger genes of interest to be added, such as the spike protein DNA. Adenoviral vectors are an example of using nature's best tool, for medical purposes as gene carriers. Although adenoviral vector has only been commercially used for animal vaccines, this technique has been applied since the 1980s for gene therapy. It has also been studied in other diseases, such as Ebola and influenza vaccines. Adenoviral vector vaccines work by injecting host cells with spike protein DNA, instead of the whole virus, as in the case of inactivated vaccines. The spike protein DNA will be processed into mRNA, or messenger RNA, by host cells, which is the instruction required for host cells to produce spike protein antigens. This method is applied by CanSino Biologics, which uses human adenovirus type 5, the most commonly studied adenoviral vector, with high effectiveness. Besides that, Sputnik V also uses adenoviral vector vaccines, but with another subtype of adenovirus, called adenovirus type 26 for its first dose, and adenovirus type 5 for its second dose. The advantage of using 2 different subtypes between doses is to reduce the potential host immune response towards the same adenovirus subtype upon second injection, due to immunological memory. Alternatively, Oxford AstraZeneca vaccine uses chimpanzee adenovirus instead of human adenovirus as vector. This is applied in consideration of the widespread exposure of human adenovirus in the community, resulting in pre-existing immunity towards human adenovirus, and a less effective vaccine response. The next type of vaccine is the relatively new method of nucleic acid vaccines, specifically using mRNA. The usage of mRNA encoding for spike protein directly, instead of its DNA, as in the case of viral vector vaccine, contributed to the rapid development of this technique. However, due to the instability of mRNA, this vaccine needs to be stored at extremely low temperature. The mRNA is also enveloped in a lipid nanoparticle to prevent degradation during its transport into host cells. And last but not least, this technique is applied by Pfizer-BioNTech. In short, these are the main types of COVID-19 vaccines that will be available in Malaysia for the months to come.